Hello aspirants, welcome back to our channel Food-Tech Online. Hope you all are doing good. So this video is for all those aspirants who are preparing for their post-graduation and want good college and they are preparing for their entrance examination. So on the subject food technology, so today is our class 2 and those who have not watched our previous video, they can watch that previous video. You'll get the link in the description box. So chalo, now let us proceed. So question number 1. Heavy use of soy products as a substitute for meat can inhibit absorption of. Options are calcium, folate, vitamin D or iron. So over here my correct option is D, that is iron. So we know that soya beans actually they contain a compound. These compounds are known as phytates. So soya bean they contain a compound. These compounds are known as phytates. So when these phytates which can bind to iron and can inhibit its absorption in the body, so what happened when we consume this product on a large scale, may this product is not processed properly or not balanced with a good iron rich product or food, what will happen, it may contribute to iron deficiency over a time. That's why heavy use of soya products as a substitute for meat can inhibit the absorption of iron. So over here my correct option is D. Question number 2. The heat of neutralization is constant for. Options are strong acid, strong base, strong acid, weak base, weak acid, strong base or weak acid, weak base. So over here my correct option is A, that is strong acid, strong base. So we know in a reaction when a strong acid and strong base it reacts, it completely dissociates in a solution to form salt and water. So since the reaction when it proceeds to the completion without any side reaction or we can say intermediate species, so the amount of heat which is released or absorbed is constant and it only depends on the number of moles of the acid or base and not on their identities. Even this is also known as the heat of neutralization. So this is when it reacts with strong acid and strong base to give salt and water. This is also known as heat of neutralization and it is around minus 57.3. I'll write over here. It's around minus 57.3 kilojoule per mole. So it's around minus 57.3 kilojoule per mole for strong acid strong base reaction at standard conditions. So over here my answer option number 2, that is it's correct. So question number 3. Which of the following food is produced by fermentation involving lactic acid bacteria? Options are beer, coffee, yogurt and vinegar. So over here my correct option will be C, that is yogurt. So the food which is produced by fermentation involving lactic acid bacteria is yogurt, and the lactic acid bacteria such as Lactobacillus bulgaricus or Streptococcus thermophilus, these are commonly used by the fermentation process to produce the yogurt. And we know that these bacteria when they ferment lactose we know that it is also known as milk sugar, so which is present in the milk, so it converts it into the lactic acid and which gives the yogurt its tangy texture or we can say a characteristic thick texture to it, tangy flavor and characteristic thick texture to it. So what are these bacteria which are involved? These are Lactobacillus bulgaricus. I'll write over here Lactobacillus bulgaricus and Streptococcus thermophilus. Okay, these are the two bacteria which help in the process of fermentation of yogurt. So over here my correct option is C, that is yogurt. So question number 4. What is the primary reason for blanching of food? Options are it prevents pest infestations, it inactivates enzyme which is found in food or cleans the food, or it prevents food from drying out. So over here my correct option will be option number B, that it inactivates enzyme which is found in the food. So the primary reason for blanching of food is to inactivate the enzymes which is found in food. So blanching it involves the immersion of food in boiling water or steam and then rapidly cooling it to the ice water, and this rapid cooling is done to stop the cooking process. So this process helps to halt enzyme activity, which may cause undesirable changes also. Okay, so over here my correct option will be option number B, that is it inactivates enzyme which is found in food. So question number 5, what is the operating principle behind oven drying for determining moisture content of foods? Options are color change is measured. Next, loss of weight represents loss of water. Change in refractive index is measured, or change in light absorbance is measured. So over here, my correct option will be loss of weight represents loss of water. So we know in oven drying method, when a sample is placed in an oven at a specific temperature for a certain period of time, what happens? There is a loss of water, means the water it starts to evaporate, and when the water content of the food it evaporates, it leads to the reduction in the weight of the food products. So the loss of the weight is then used to calculate the amount of moisture content which is present in that food products. So this method is basically based on the principle that the weight of loss during drying corresponds to the amount of water that was originally present in the sample. That's why there is a loss of weight which represents the loss of water. Over here my option number B is correct. So question number 6. Which of the following packages is an example of aseptic packaging? Options are Tetra Pak drinking boxes, paper bag, milk carton or plastic bag. So over here my correct option will be option number A, that is Tetra Pak drinking boxes. So the Tetra Pak drinking boxes, it is an example of aseptic packaging. So aseptic packaging actually it involves sterilizing the packaging material and the contents separately, and then filling the sterilized contents into the sterilized package in a sterilized environment. And this process actually ensures that the packaged food product is free from harmful microorganisms, which allows to store at a room temperature for extended period without the need for the refrigeration or preservatives. So this Tetra Pak, it is well known for, it is actually a well known brand which that utilizes the aseptic packaging for various beverages. So these can be used in the milk or other liquid products. So over here my correct option is Tetra Pak drinking boxes. It is an example of aseptic packaging. Next question number 7. Which gas causes fruits to ripen? Options are carbon monoxide, propane, ethylene or nitrogen. So over here my correct answer will be the gas that causes fruits to ripen. It's ethylene gas. Correct option is option number C. So this ethylene, it is a plant hormone that occurs naturally in fruit, and it involves in the regulation of various physiological processes like the ripening process. So over here my correct option is C. Next, question number 8. Which of the following chemical is a solvent which is used for testing fat content? Options are hydrochloric acid, sodium hydroxide, amylase or ether. So over here correct option is D, that is ether. So the chemical which is commonly used as a solvent for testing of fat content is ether, and we know ether it is often used in a method which is called Soxhlet extraction, and which is commonly used technique for extraction of lipids, say fats, and from solid food samples. So over here my correct option is number D, that is ethylene. So I'll write over here the method, that is Soxhlet extraction. Okay. So correct option is D, that is ether. Question number 9. Which of the following ingredient in chocolate milk comes from seaweed? Options are carrageenan, glucose, sucrose or cocoa. So over here my correct option will be option number A, that is carrageenan. So the ingredient in chocolate milk that comes from seaweed is carrageenan, and we know this carrageenan it is used as a common additive for a thickening and a stabilizing agent in various food products, for example this chocolate milk, and it is derived from the red seaweeds. So it is derived from basically from red seaweeds. Okay, red seaweeds, it is basically derived from red seaweeds, and it is added to chocolate milk to improve its taste, texture or creamy consistency to the milk. So over here correct option is A. Question number 10. Which of the following food is rich in omega-3 fatty acids? So options are lard, fatty fish, butter and olive oil. So over here my correct option will be, that is fatty fish. So the food which is rich in omega-3 fatty acid, it's fatty fish, and this fatty fish actually, such for example we'll say mackerel or salmon, these are the excellent sources of omega-3 fatty acids. So particularly they'll have For example, EPA, that is eicosapentaenoic acid, or DHA, that is docosahexaenoic acids. So these fatty acids, they have numerous health benefits to our health. Okay. So over here my correct option will be B, that is fatty fishes. So next, question number 11, which microorganism is commonly associated with the fecal contamination? So options are Clostridium botulinum, Campylobacter jejuni, Bacillus cereus, and Trichelina spiralis. So over here my correct option is option number B, that is Campylobacter jejuni. So the microorganism which is commonly associated with the fecal contamination is Campylobacter jejuni. So this bacterium, basically it is a major cause for the foodborne illness worldwide, and it is often transmitted through the contaminated food or water, or through contacted or infected animals, or with their species feces. So it basically causes a disease that is known as campylobacteriosis. So this campylobacteriosis is the illness which is caused by the Campylobacter jejuni. So over here my correct option is B, that is Campylobacter jejuni. Next, question number 12. Which industrial processing method is most effective for making dried potato flakes? Options are drum drying, spray drying, sun drying, and osmotic drying. Dehydration. So over here correct option is A, that is drum drying method. So the industrial method which is most effective for making dried potato flakes is the drum drying method. In drum drying method, actually it involves the spreading of the thin layer or the mashed or purees of the potatoes on the hot surface of the rotatory drum, and as the drum it starts to rotate, the moisture which is present in the potato, it starts to evaporate, and it leaves leaves behind the dried potato flakes. So the drum drying, it is particularly effective for producing potato flakes behind. It allows for the precise control over temperature and drying time, resulting in a consistent product quality. Additionally, over here I can say one more thing, that it can handle a large scale production efficiently, whereas in sun drying it cannot be handled at a large scale, whereas drum drying it can be handled at a large scale. That's why it's used as at an industrial level for producing the dried potato flakes. Over here my correct option will be A, that is drum drying method. Next, question number 13. What additive in salt prevents the thyroid condition known as goiter? So options are sodium caseinate, titanium oxide, coccineal extract, and potassium iodide. So over here my correct option will be D, that is potassium iodide. So the additive in salt that prevents the thyroid condition known as goiter is potassium iodide. We know that iodine deficiency, it leads to the cause of goiter, and a condition which is characterized by an enlargement of the goiter thyroid gland actually. So this adding potassium iodide or other iodine compounds to the table salt, which we say as the iodized salt, which helps to prevent this iodine deficiency, and even it reduces the risk of the goiter. That's why over here my correct option is D, that is potassium iodide. Next, question number 14. Which of the following microorganisms cannot tolerate oxygen? So options are Clostridium botulinum, Staphylococcus aureus, Penicillium roqueforti, or E. coli. So over here my correct option will be option number A, that is Clostridium botulinum. We know this Clostridium botulinum, it is an anaerobic bacteria, meaning it cannot survive in the presence of the oxygen. So over here Clostridium botulinum, it is an anaerobic bacteria. Okay, anaerobic bacteria. So over here my correct option will be option number A, that is Clostridium botulinum. Question number 15. Which of the following method is a quick test for the sugar content during the early stages of brewing process of beer? So options are hydrometer, backup test, wet ashing, or Soxhlet extraction. So over here my correct option will be A, that is hydrometry. So basically hydrometry involves the use of a device which is called hydrometer, and it is used to measure the specific gravity of a liquid, which gives the indication to the sugar content. So over here my option number A is correct, that is hydrometry. Next, question number 16. Which of the following processing method involves the heating of food at a high temperature for short period of time in order to reduce the risk of food poisoning? So options are blanching, irradiation, pasteurization, or home heating. So over here correct option is C, that is pasteurization. So the process or the method that involves the heating of food at a high temperature for short period of time, we often say it by the name HTST, in order to reduce the risk of food poisoning, that is known as pasteurization. So during pasteurization pasteurization food or the beverage, these are heated to a specific temperature for a defined period of time, usually below the boiling point, and it is used to kill harmful bacteria or pathogens while preserving the sensory nutritional qualities of the food products. So over here correct option is C, that is pasteurization. Next, question number 17. What causes sliced apples to turn brown? So options are A, caramelization, staling, enzyme activity, protein degradation. So correct option over here is C, that is enzyme activity. So the sliced apple, when it turns brown, the enzyme activity occurs. So when we actually cut an apple, what happens? The cell of the apple, it ruptures or it get damaged, and it get in contact with the oxygen. What happens? It triggers a series of reaction, primarily the oxidation, then phenolic compound present in the apple, resulting in the formation of brown pigments called the melanins. And this process is commonly known as enzymatic browning reaction. Okay, so over here correct option is C, that is enzyme activity. Next, question number 18. Which of the following does not have antimicrobial activity? So options are chlorophyll, organic acids, spice extracts, or the hydrogen peroxide. So over here correct option is A, that is chlorophyll. So chlorophyll does not have antimicrobial activity. So while organic acid, the spice extracts, hydrogen peroxide, these are known for their antimicrobial properties. Rather chlorophyll, which is responsible for the green color in the plant, so it does not have any significant antimicrobial activity. Okay, so our correct option over here is my chlorophyll. Question number 19. From which plant source is gluten derived? Options are soya beans, corn, rice, and wheat. So correct option over here is my D, that is wheat. So gluten, it is derived from wheat, and it is a composite of protein which is found in the wheat and its related wheat products like say rye or barley. So gluten, basically it is responsible for providing the elasticity of the dough, which gives bread its characteristic chewiness, or it helps to rise during the baking process. So over here my correct answer is number D, that is wheat. Next question number 20. HDLS are synthesized in, options are blood, liver, intestine, and pancreas. So over here my correct option will be number B, that is liver. So this HDLS basically it stands for high density lipoprotein, and these are synthesized basically from the liver. So this liver is the main organ from which is responsible for the synthesis of HDL particle, and this HDL, it plays a crucial role in the transportation of cholesterol from the peripheral tissues back to the liver. Okay, where it can be metabolized, excreted, or from where it excreted from the body. Okay, so over here my correct option will be that is liver. Question number 21. What for the recovery of citric acid after fermentation, calcium hydroxide is added to the slurry to precipitate calcium carbonate, precipitate calcium citrate, precipitate calcium phosphate, or precipitate calcium sulfate. So over here correct option will be number B, that it precipitates calcium citrate. So for the recovery of this citric acid fermentation, calcium hydroxide, it is added to the slurry to precipitate this calcium citrate. Option number B is my correct option. So question number 22. Alligar is a type of vinegar which is produced from, options are fruit juices, malted grains, ethanol, and ale. So correct option over here is my D, that is ale. So alligar, it is a type of vinegar which is produced from ale, and it is a specific type of vinegar that is traditionally made from the fermentation of ale, a type of beer which is allowed to undergo acetic acid fermentation to convert the alcohol into vinegar. So correct option over here is my D, that is ale. So question number 23. The support material for immobilization of cells for Bacillus subtilis is, options are ion exchange resins, gelatin, anthracite, agarose, and carbodimide. So over here correct option is D, that is agarose and carbodimide. So the immobilization of cells, basically it involves entrapping or attaching of cells onto the solid support matrix and allowing them to remain in their fixed positions while being still able to carry out their metabolic functions. So agarose, it is a polysaccharide which is derived from a seaweed, and it is commonly used for the support matrix due to its biocompatibility and its ability to form the gel. Whereas I can say this carbodimide, it is a chemical compound which is often used to cross-link the agarose matrix and the cells. It provides the stability to immobilize the cells. So correct option over here is D, that is agarose and carbodimide. Next. Next, question number 24. Which of these enzyme contains a zinc ion? Options are carboxypeptidase A, phosphorylase, B kinase, tyrosine hydrolase or hydroxylase or phosphodiesterase. So over here my correct option will be option number A, that is carboxypeptidase enzyme. So this carboxypeptidase enzyme, it is also known as metalloenzyme, means it requires a metal as a cofactor for its activity. In the case of this carboxypeptidase A, the metal ion or the cofactor is the zinc. Over here, correct option is A. Next. Next, question number 25. Sterilization by radiation is called as, options are cold sterilization, radioisotopes, radiotype or red sterilization. So I want you all to comment me down the correct option for this question. Okay. So, and yes, if you have still not installed our app FoodTech Online, then you can install this app. The link is in the description box for you all. This app is beneficial for all those aspirants who are preparing for any food related examination. Okay, they can practice questions over here and they can enhance their preparation. This we have completed few important post-graduation level entrance examination questions. Further questions you can practice in our app. And if you like this video, you can share with your friends, subscribe the channel for more such videos. Till then, thank you, enjoy learning.